In this podcast, we're going to look at proteins involved in transmembrane transport. So these molecules, these proteins, facilitate the transport of molecules such as ions that cannot diffuse across the cell membrane. There are several examples of such transmembrane transport proteins found on the parietal cell that are important for the secretion of hydrochloric acid. And essentially there are three classes of these transmembrane proteins. For instance, there are the ion channels that facilitate the movement of potassium ions at the apical membrane. Transporter proteins, which facilitate the transport of chloride to the apical membrane, or that couple the movement of chloride ions with bicarbonate ions at the basolateral membrane. And finally, there is the third class of transport proteins which require energy and where ATP hydrolysis is required. Now it should be noted that these transmembrane transport proteins are all integral membrane proteins, and also that these transport proteins are not just important for secretion of hydrochloric acid by the parietal cell. These proteins are found on many different types of cells. There are many hundreds of different transport proteins. Some have very specific functions for only specific cell types, while others may have very general roles and can be found in many different cell types. So these transport proteins are really very important for basically all cell types in the human body. Now let's first look at ion channels. So ion channels are conceptually perhaps the easiest to understand. So essentially these are just tunnels through the lipid bilayer, through the membrane, allowing diffusion of ions down the gradient. And this is just by simple diffusion. The movement of ions is really remarkably fast, about as fast as the theoretical models predict, based on free diffusion. But despite this rapid movement, these ion channels can be highly selective. So for instance, a typical potassium channel can achieve 10,000-fold selectivity compared to the movement of sodium chloride, despite the great similarity of sodium and potassium ions. Ion channels can either be open or closed, and this is facilitated by a so-called gate. And there are two types of ion channels depending on how the gate is regulated. They are either voltage gated ion channels or ligand gated ion channels. So voltage gated ion channels are regulated by the membrane potential, and so when the gate is closed, ions cannot move through the channel, but when the gate is open, depending on the change in the membrane potential, ions can diffuse through. The second type of ion channel is the ligand-gated ion channel. And so here, in the absence of ligand, the ion channel is closed, but when the ligand, the small molecule ligand, binds to the ion channel, the channel is opened and the ions can move through the channel by free diffusion. And so these types of ion channels can act as receptors, either of electrical signals or of small molecules, and they're really important for the function and signaling in the nervous system. Now there are many different ion channels present in human cells, And the diversity of ion channels is illustrated by these various structures that are available. So for instance, here is an ion channel that is ATP gated, so ATP is required. So you're looking here down the channel, and it's composed of 3 identical subunits. So it's in fact, it's a homotrimeric complex. The second example here is a voltage gated potassium channel. It's composed of 4 identical subunits in different colors, and again you're looking down the channel. So this is a homotetrameric complex. And a final example that I found is, for instance, this glutamine gated chloride channel, which is a ligand gated channel composed of 5 different identical subunits that form a homopentameric complex. And again, you're looking down the channel. Ion channels are a really important class of drug targets. So apart from GPCRs, which are the largest class of drug targets, ion channels, that is ligand gated and voltage gated ion channels combined, are the second largest class of drug targets. So some drugs that target ion channels have been around and used in the clinic for more than 50 years. Others have entered the market or the clinic only more recently. Some target voltage gated channels, some ligand gated channels, and so combined this is a very important class of drug targets. The second class of membrane transport proteins are the so-called solute transporters. And again, there are different types of solute transporters. So here in this scheme is the uniporter. We'll talk about the antiporter and symporter in a moment. So a uniporter will bind a molecule that will be transported on one side, and then some kind of conformational change is required for the molecule to be transported to the inside of the cell. And so this is the sort of difference between solute transporters and ion channels. So ion channels have a form a tunnel through which ions can just simply diffuse. But in the case of solute transporters, these proteins are really different, and some kind of conformational change is required for this transport to occur across the membrane. So this is an example of a uniporter where the transport is driven by a concentration gradient. And just as we've seen in the case of ion channels, these solute transporters can also be very selective in the recognition of ions and molecules that are transported. In addition to uniporters, which facilitate the transport of a single molecule down the concentration gradient, there are also solute transport proteins that couple the movement of one molecule to the movement of a second. So if this movement is going in the same direction, it is called a symporter, which is here shown on the right schematically. So in this case, movement of one molecule against the gradient is facilitated by the movement of another molecule, an ion, in the same direction down the gradient. So this movement of the ion down the gradient is energetically favorable, and it provides the energy for the movement of the other molecule against the gradient. So an example of a symporter is, for instance, the sodium glucose importer that is found in intestinal cells, where the uptake of glucose against the gradient is facilitated by movement of sodium ions down the gradient. So when the coupled movement of one molecule is going in the opposite direction as movement of the second molecule, we're talking about an antiporter mechanism. So an example of an antiporter is found in a parietal cell, for instance the bicarbonate chloride antiporter. So in this case, again, movement against the gradient of one molecule is facilitated by the movement of a second molecule down the gradient. There are really many solute transporters encoded by the human genome. There are more than 350 different of these solute transport proteins. Some of these solute transport proteins are important because they are facilitating the uptake of toxins, or they are important for the intracellular uptake of drugs. So this is an area that's actually relatively unexplored. Many transport proteins are also related to human disease, and this is just a very small number of examples of mutations that occur in solute transport proteins. So for instance, a mutation in this thyroid hormone transporter can lead to a varying degree of psychomotor retardation, speech and movement. Mutations in these transport proteins are associated with Bartter syndrome and Gitelman syndrome. So these are syndromes that are essentially kidney disorders, which can also arise from a mutation in a chloride channel. And just a final example, mutation in this transporter, SLC12A6, results in Andaman syndrome, which is characterized by motor and sensory neuropathy. So for instance, there's pain insensitivity in this syndrome. So altogether, solute transporters have a very clear pharmaceutical relevance as well as a medical relevance. The third and final class of transmembrane transport proteins are two groups of transport proteins that require ATP in order to transport molecules against the gradient across the membrane. So there are two types of these ATP-dependent transporters. The first is the ATP-dependent ion pumps, which are particularly relevant in the secretion of hydrochloric acid by the parietal cell. And the second group are the ABC transporters. So both types of transport proteins facilitate transport against the gradient across the membrane, and they use ATP to provide the energy required. So in both cases ATP is hydrolyzed in order to facilitate transport against the gradient. Of note is that the rate of transport is significantly lower than that observed in the case of the ion channels and solute transporters. So the ABC transporters is a large group of proteins characterized by 6 transmembrane alpha helices, and these transport proteins are involved in a variety of processes. several ABC transporters are associated with human disease. So, for instance, cystic fibrosis, which is the most common genetic disease in humans, is caused by a mutation in the CFTR gene, which encodes a chloride transporter. Another example is cytosterolemia, which is caused by mutations in either the ABCG5 or the ABCG8 gene, which both contain an ABC transporter, and these are involved in sterol and cholesterol transport. And some of these transporters are also important, from a pharmaceutical perspective. And they are important, for instance, in drug resistance. So the P-glycoprotein is a prime example. So this protein is implicated in the export of drugs. So they prevent drugs to reach their target, and this is particularly relevant in cancer and antiviral therapy. So the ion pumps, they are characterized by a multitude of alpha helices that cross the membrane. Usually there are different subunits, two subunits that work together as a single functional unit. So in case of the gastric proton pump, the alpha subunit contains 10 of these transmembrane helices, and a large intracellular domain contains the ATP hydrolysis domain. So by contrast, the beta chain of the gastric ATPase contains only a single transmembrane helix and a relatively large extracellular domain. Here I show the structures of three related ion pumps. So these all contain a bundle of transmembrane helices and cytoplasmic domains which are involved in the hydrolysis of ATP that provide the energy for the movement of ions across the membrane. So the gastric proton pump here is composed of two subunits, and it exchanges the two protons for two potassium ions, or only a single proton for a single potassium, depending on the pH per ATP hydrolyzed. And so important is that it is a electroneutral process. And of course the gastric proton pump is an important drug target. It's inhibited by proton pump inhibitors, which are used in for the treatment of gastric ulcers, and we'll talk about that later on. The sodium-potassium ATPase is perhaps the best known proton pump, the best characterized proton pump. It contains three subunits, and it's different in the compared to the gastric proton pump in the sense that it is not electroneutral, so it exchanges 3 sodium ions for 2 potassium ions per ATP hydrolyzed, so the ion transport is not electroneutral. So the sodium-potassium ATPase is also a drug target in heart failure by digoxins, for instance, which bind presumably in this site here. And a third well-known ion pump is the calcium-sodium ATPase, which is an intracellular ion pump located on the membrane of the endoplasmic reticulum, and this ion pump is very important for muscle contraction. So to sum up this and a previous podcast, we can classify membrane proteins in at least 2 different ways. Firstly, we can classify membrane proteins based on the manner in which the proteins interact with the membrane, and so we can distinguish in this way integral membrane proteins, which or also known as transmembrane proteins, and so we've seen several examples, for instance G protein coupled receptors, or proteins involved in transmembrane transport. Another class of membrane proteins are lipid modified membrane proteins, for instance the G alpha and G gamma proteins that are involved in signaling. And so these proteins contain these lipid anchors which are inserted in the membrane. And a third class of membrane proteins are these peripheral or accessory membrane proteins. So these are the proteins that do not directly interact with the membrane, but indirectly through interactions of membrane-associated proteins. An alternative way to classify membrane proteins is by function. So we can distinguish, for instance, proteins involved in signaling, including receptors, or we can distinguish proteins involved in the transmembrane transport of molecules, proteins involved in attachment and migration, for instance proteins that are involved in the formation of tight junctions, of adherent junctions, and we have proteins that have other roles. For instance, we've talked about the mucins that provide a protective function